So now that we've gone through the generalities of the nervous systems, how it generally functions, now we're going to focus on a specific branch here. We're going to focus on the sensory division of the nervous system, or the afferent division. Afferent's bringing information from the outside of the peripheral back into the central nervous system. And so the function of this is basically we have changes taking place, either outside the body or inside the body, and these are changes that typically we have to respond to. And so that's the job of the sensory nervous system, is to detect these changes and bring that information to the central nervous system. Then the central nervous system can make a decision on what to do, and then use the other branch that we're going to talk about later, either the autonomic or the motor nervous system, bringing information out for a response. Okay, but the job of the sensory is to detect changes taking place and relay that information. And so how this takes place is there are multiple parts to the system, and it starts off with sensory receptors. Okay, because these are changes taking place, and there's different stimulus for changes. The vision, when you're looking at light energy, is different than auditory information that's coming in through sound or so forth. Okay, so there has to be specific sensory receptors for these different stimuli that we're trying to detect. Okay, and what the sensory receptors function is basically taking whatever stimulus, say for the eye, it's photo energy or photo waves, and taking that information, because the light doesn't go directly to the brain. Basically, how does the brain operate? It operates through signaling through the nervous system through action potentials. So what the sensory or receptors function is to do is to transduce. Okay, transduction means taking whatever stimulus, in this case photo energy for vision, and turning it into a nervous impulse or electrical impulse. Okay, so that's the job of the receptor. Okay. And where we find the receptors are going to be here at the dendritic end of the sensory neuron. Okay. The sensory neurons, once the receptors have transduced the information, the sensory neurons, if there's enough stimulus to cause an electrical impulse, the sensory neurons are going to carry that information up to the central nervous system. And usually this isn't just a one neuron going all the way up, it's a multiple neuron pathway. But these sensory neurons are going to carry the information up to the central nervous system. All right, and then the central nervous system can perception and interpretation. So if we're looking out and there's a bright light that flashes in front of us, the sensory receptor is going to take that light, transduce it to an electrical impulse. Right, then those impulses are going to be sent up via the sensory neurons, and then the brain gets that information. Hey, I'm getting information from my sensory neurons for location for light, and the brain can put a perception to it. Okay, so for this to work, for the sensory system to work, we have to have all three parts working. The sensory receptors have to be operating, sensory neurons have to operate, and the portions of the brain, because you can see there are different portions of the brain that are dedicated to the different sensories or different stimuli. Right? We're going to talk about cutaneous, those that lie on the skin, all right, or throughout the body. Those go to the somatosensory cortex. If what we were talking about was vision, that information would have been brought back, carried back to by the sensory neurons to the visual cortex. Okay, so if you have a lapse or a breakdown in either the receptor, the sensory neurons that bring it up, or in this case, say the visual region, okay, you won't be able to perceive or have a dysfunction in perceiving the different stimuli or different changes taking place. Okay, let's look at this setup here. Okay. This is the beginning, the sensory receptors. Okay. And where they are located, the receptors are basically the dendritic end of the first sensory neuron. And the way the setup of these can be multiple. Here we actually have the dendrite of the sensory neuron. Okay, and it's encapsulated in this specialized capsule. Okay, but the receptor is the dendritic end of the sensory neuron, and it's this specialized capsule. Okay, so that is one setup for these sensory receptors. Another setup for these is, here's a sensory neuron, here's the dendritic end, but instead of the receptor being part of the actual neuron, the sensory neuron, the receptor is a specialized epithelial cell. And you can see they are closely associated with the sensory neuron. Okay. And so we have the specialized epithelial cells, and they actually operate similar to a neuron. Okay. You can see they get the stimulus, causes the cells to be activated or depolarized, and you can see these specialized epithelials release neurotransmitter or chemical signal just like a neuron would, but they are not neurons. They are actually made from specialized epithelial. Okay, but they act very similar. That sending of the transmitter will activate that neuron, and then the neuron can send the information, right, via the sensory neurons up to the brain. Okay, the one not shown here is, instead of having this dendrite in a specialized capsule, you could have dendrites. Okay, these would be the dendrites here. These would actually be the receptor end. Okay, these are free nerve endings. Okay, so the receptors are actually on the dendrite itself, so on the neuron itself. Okay, so these are the different setups. So when we go through the different sensories, you can see there are different setups for the different senses. Okay, some specialized ends of the dendrite, the free nerve endings, and then there are some senses that operate through specialized epithelial, or that's where the stimulus gets transduced. Okay, but what's taking place at these receptor ends is the stimulus is being transduced, converted from whatever stimulus it is into an electrical impulse. So for these sensory receptors, these ones that transduce that stimuli into electrical signals, we can classify them several ways. One way is in where the stimulus comes from. Okay, if the stimuli is from inside the body, say on receptors that pick up blood pressure, changes in blood pressure that are going on in our baroreceptors, right? That information is coming from inside the body. Right? It's coming from the blood vessels inside our body is where these baroreceptors reside, and they are picking up the stimuli from inside the body, so they are interoreceptors. Okay, exteroreceptors, on the other hand, they receive stimuli from outside the body. Just going from our example with light energy. The photoreceptors are your eyes. The light energy is coming from outside the body. If there's a flash of light happens in front of you, your eyes, your photoreceptors pick up this flash of light and can transduce that energy and send it back to the brain. And photoreceptors are picking up the stimuli from outside the body, so they are exteroreceptors. Okay, we can also classify on what stimulates these receptors. What does it stimulate that activates these different sensory receptors? If it is chemical, then they are chemoreceptors. A classic example is taste, gustation. Gustation the receptors for gustation respond to chemicals in the mouth. Okay, those receptors on the taste cells in your taste bud respond to different chemicals. So for sweet, say glucose would be a stimulus for a sweet receptor. Or for salty taste, it would be sodium ions. Okay, for salty. But they are chemicals. Chemicals are the stimulus for activating these receptors. Another type of stimulus is mechanoreceptors. Okay, this is the receptors respond to distortions in the membrane of the cell. Inside your ears, you have these sensory cells, okay, that have basically like hair-like projections, and it's going to be movement of fluid across these hairs that will actually cause these to bend. And the bending causes distortion in the membrane, and that bending will activate these receptors. So it's the distortion in the membrane is the stimulus in this case for mechanoreceptors. For your eye, it's photo. Okay, photo waves have energy, and they activate photoreceptors that are in the cornea in the back of your eye. Or sorry, the retina in the back of your eye, not your cornea, your retina. Okay, so we'll get into the details of that later on, but the retina is the layer in which these photoreceptors reside, and light coming into the eye will activate those receptors. Okay, and then there are thermoreceptors. Thermoreceptors respond to warm and cold. Okay, located near your skin. When you touch warm water of a spa, that would activate the warm receptors. When you touch ice cold water, that would activate the cold receptors. Okay. And lastly, there is one stimuli that is not located here, and that would be nociceptor. We know what the stimulus is for nociceptor. Those are the receptors for pain. So the receptors that stimulus is pain are nociceptors. And pain can come from many different types of stimuli. So there are receptors, different nociceptors for different kind of stimuli, like high heat, not just warm, but high heat, or extreme cold, pressure, and also chemicals. Chemicals that from tissue damage. When your tissue is damaged, it releases chemicals, and nociceptors many times have sensory receptors that respond to those chemicals, so we can detect damage taking place in the tissue. And if you remember from our nervous system lecture, glutamate. Okay, glutamate is one of the main neurotransmitters in the sensory pathway up to the brain for nociceptors. Remember, PCP blocked glutamate. So you saw talked about people with that were on PCP didn't feel any pain. They basically pull their shoulder out of socket and not even feel the pain because they are blocking that glutamate. So they're blocking the response. Now let's look at some of the processes or the way in which these receptors operate. Okay, some of these receptors, they can go through what's sensory adaptation. Okay, almost can stop being aware, stop responding to the stimuli. That would be sensory adaptation. Okay. So our phasic, if your sensory receptor is a phasic receptor, okay, these are considered fast adapting. So what happens in these receptors, so these are all action potentials by the sensory neuron. And if you remember going back from our nervous system lecture, okay, how is stimulus strength coded? It has to do with frequency. Okay? You apply a stimulus, you get a higher frequency of action potentials firing. Okay? And that's sensory neuron. Say you sit down on the chair in class or at your computer. You feel the pressure on the back of your legs from the chair as you sit down. And so that neuron, that pressure neuron, those neurons associated with the pressure receptors fire. But as you continue sitting, what happens? Do you constantly think about, hey, there's pressure on my legs, there's pressure on my legs? No, you're focusing on what's going on the computer. You've basically stopped becoming aware of the pressure on your legs. And that is because when you, with phasic receptors, the initial stimulus causes them to fire, and your brain can interpret, your brain's getting lots of signals from those pressure receptors saying, hey, there's pressure on my legs. But as the stimulus is continually applied, those receptors slow down the response to the stimuli. And you see the frequency becomes much slower. Lots more time in between the frequency of action potentials. And your brain gets this, and your brain interprets, hey, no more pressure there. Because of this decreased firing rate. That's what happens in phasic receptors. Many times odor, touch. After constant stimulation, those receptors for these start to dampen down. So it allows us to kind of filter out unimportant stuff. When we're sitting at the computer, do we need to know that we have pressure on our legs? No, it's unimportant at the time. We want to focus on what's happening in the computer. But there are things where we don't want phasic receptors. We would want a tonic receptor. Tonic receptor is slow adapting or non-adapting, and you can see differing from what we see up here. The stimulus is applied, and as we continually, the stimulus is still there, what do you see? You don't see a slowing down of the action potentials. So your brain is constantly, hey, the stimulus is here, the stimulus is here. The classic tonic receptors are going to be your pain receptors. Usually it is hard to get pain out of your when you've hurt something, it's hard to get pain out of your mind because they are tonic. They are constantly sending signal, hey, there's damage here, there's damage here. And that's important because that is not unimportant information. We need to know pain because they are responding to typically damage that is happening to the cells. So nociceptors are tonic. They don't slow down. You get a constant, hey, there's pain here, there's pain here. That's letting your body know you need to take some action to relieve that pain. In the nervous system lecture, we talked about graded potentials and how graded potentials, if strong enough to reach threshold, cause an action potential. So receptors, they behave similar to the dendrites, where we see those graded potentials in that the stimuli there, whatever the stimuli is, for the receptor causes generator potentials, causes those graded potentials. And just like the graded, they can be graded. They can be at different varies. With smaller stimuli, you get a small generator potential, graded potential, and with the bigger, you get more. But if we have enough stimulus, it allows us to get to threshold, and that stimulus can cause an action potential in that sensory neuron. So like in touch, your Pacinian corpuscles of your skin, very light touch would cause a small generator potential, where an increased touch, increased pressure would cause a bigger generator potential that might elicit the action potential. So again, it is a graded response. So also in our nervous system, we talked about how stimulus strength is coded. If you remember, if we have an action potential, it's all or none. So the action potential is going all the way or none at all. So how do we code for, hey, light touch versus a strong touch? And that code is through frequency. How many action potentials are being sent by, in this case, the sensory neuron. Okay. And you can see, example here is we see light touch, a weak stimulus. Right? That would be coded purple here, this light, smaller purple, that's a stimulus strength. You can see the receptor potential, the greater depolarization it causes, and what does that do for action potential wise. You can see the frequency. Each of these spikes is an action potential by the sensory neuron. You see frequency, decent but not super fast, not high frequency, where when you see a strong stimulus here, okay, you see it coded here, and what's the receptor potential, that generator potential it produces at the dendrite, be much larger. And what do we see with action potentials sent by this neuron? You can see the frequency is much more frequent. Okay, so the strength of the signal is coded by the frequency. In terms of the sensory receptors, there's a term called law of specific nerve energies, and that deals with each sensory receptor. They have a particular stimulus that elicits a response, elicits those depolarizations from them. You can see for photoreceptors, those ones in the retina of your eye, okay, vision, and what's the modality that actually elicits a response in those receptors are photons of light. You see the different elicits are chemoreceptors. Okay. Taste, smell, pain are all examples of chemoreceptors. You can see what are the modalities. Okay. What's going to elicit a generator potential in those sensory receptors. Okay. And the stimulus to which a receptor responds to is termed the adequate stimulus. And that refers to what elicits a response in the different receptors. So our senses can be classified into two categories on kind of where the sensation is coming from and located, where the stimuli is coming in. We have our general senses, and those would be stuff like what we're going to talk about here, our cutaneous sensations, right? Proprioception. We have sensors in our joints to let us know where our joints are located or what angle our joints are located. And these are all coming throughout the body. They're located throughout the body, and these would be termed our general senses. The other are our special senses, and these are sensations that are located specifically in the head region. Stuff like our taste, our vision, our equilibrium, our auditory, our hearing. Okay, these are all located in the head region, and these are our special senses. So we're first going to hit on one of the general senses, the cutaneous sensation here, and also termed somatosensory sensation. And this kind of gives you a clue on where this kind of sensation, these general senses, go to in the brain. If you remember, there are different regions of the brain devoted to different senses. And in this case, our general senses, the information is picked up by the sensors in the skin and brought ultimately to the somatosensory cortex in the brain. Our cutaneous sensory receptors. We have mechanoreceptors, we have thermoreceptors, and we have nociceptors. Those involved in touch and pressure. You might remember from your anatomy days in your tegumentary the Pacinian corpuscles. Ruffin endings, Meissner corpuscles, these are all mechanoreceptors because distortion, pushing down, distortion of the membrane results in sensation of touch and pressure in this case. For thermoreceptors, these are stimulated by warm and cold, not extreme warm, not hot, and not extreme cold. These are within these specific ranges. Because once we get out of these ranges, the sensation would be from the nociceptors. Those extreme ends of hot and cold are stimulate nociceptors, but those within that warm and cold range stimulate the warm and cold receptors, the thermoreceptors, just underneath the skin. So our first of our cutaneous sensations, let's look at those involved in basically touch and pressure. You can see we got different setups of these. We have free nerve endings, Merkel disc, Ruffinian corpuscles, Meissner corpuscles, Pacinian corpuscles, right? And these are all located basically at the, if you remember, we have the two layers. We have the upper, okay, more superficial is the epidermis, and then the dermis is just underneath that epidermis. Okay? And these receptors are located, basically there is the Merkel disc at the base of the epidermis in that bottom, the deepest layer of the epidermis, but then the rest are located throughout the dermis. Okay, but you can see the different sensations for the free nerve endings, stuff like light touch. That was kind of that one we saw with the frequency of coding. Those was a free nerve ending, filling for touch. Merkel disc, sustained touch in depth. The Ruffinians are going to be involved in when you're getting stretch. You're getting pressure put on and it's stretching the skin. That would be sensed by the Ruffinian corpuscles. And then slow vivations, change in textures, are these Meissner corpuscles that are just located deep to the dermis. And then the deep pressure, okay, are those Pacinians that are located deep in the dermis. So those would be for touch. Those would be mainly mechanoreceptors involving these different sensations. So just under the skin, we also have cold receptors. Those are basically, the stimulus for them is cold on the skin. Okay, they're going to be stimulated by cold, so that when cold hits, the frequency of action potentials in that sensory neuron is going to be much more frequent. In a warm, the frequency is going to be much more drawn out, the frequency of action potentials. Okay, so the receptors respond to cold and inhibited by warm. They also are responsive to, we saw on the previous slide, that menthol. When you have menthol, you feel the feeling of cold, okay, and the temperature range, 8 to 28 degrees Celsius. Okay, we'll see there are some overlaps between the warm and cold and where they get stimulated. But one thing I want to point out is, these cold, warm receptors and nociceptors throughout are, the receptors for them are these transient receptor potential channels, TRP channels. Okay, you don't need to know all these, but you do need to know transient receptor potential, because these channels, when stimulated, are open, and they are ion channels for sodium and calcium. It is what's going to induce the depolarization in the sensory neuron. Okay, so TRP channels, transient receptor channels, when stimulated, open, and they are channels for sodium and calcium. And you see there's a diverse bunch of these TRP channels throughout the body, stimulated by various stimuli. So similar to the cold receptor, warm receptors respond to changes of temperature taking place around the skin. Unlike the cold receptor, the stimulus for them are the warmer temperatures. As temperature increases, we can see the signaling in those warm receptor neurons increases. So you can see at 32 degrees, the frequency of action potentials in the sensory neuron is much less frequent. But when we increase up to 39 degrees, you can see the difference. The frequency of action potentials in those sensory receptor neurons, those neurons that contain the warm receptors, the frequency is much greater. This is different from extreme temperatures, because remember, extreme temperatures, heat, really hot, is detected by the nociceptors, okay, the pain receptors. And that's what we see here with the not warm but hot receptors. Okay, the warm receptors respond within range of temperatures here, but once we get to extreme temperatures, there are receptors, hot receptors for pain, okay, that are going to respond to the extreme temperatures. These extreme temperature receptors are also transient receptor potential channels. So when we get high heat, they're going to respond by opening up, and again, they serve as sodium and calcium channels when responding. So in the sensory nervous system, the sensors are out in the peripheral picking up and detecting these changes in the stimuli, and that information gets picked up, but ultimately it has to go to the brain to get perception. And so we have these sensory pathways that are involved in taking the information from the sensory receptor and bringing it to the brain. And usually these pathways are multi-neurons. There's multiple neurons. It is not the sensory neuron that picks up the sensation, brings information all the way to the brain. Usually there are multiple neurons involved in these pathways. Usually 3, sometimes only 2, but usually it's a multi-neuron pathway bringing the information to the brain. And one of those pathways or tracks that we see in the spinal cord is the anterior spinothalamic or lateral spinothalamic, and you can see the cutaneous information or sensation that is brought up there. For crude touch and pressure, we're coming up the anterior pathway. Okay, that would be this pathway. So the neurons involved in bringing sensation for crude touch and pressure, they would be ascending. It would be an ascending track in the spinal cord. So these neurons would be the axons climbing up the spinal cord headed towards the brain. Okay, the lateral spinothalamic is that pain and temperature from cutaneous. Okay, and that would be the neurons, the axons for the neurons here would be climbing up the spinal cord in this region here. So let's take a look further at the pathway here, but this, through the spinal cord region, these would be the regions in which the neurons are climbing up and trying to bring the information to the brain. So let's look at this anterior and lateral spinothalamic pathway in a whole. Remember, there's multiple neurons that make up these sensory pathways. So we have a first order neuron, second order neuron, and third order neuron. So we have 3 neurons that make up this pathway that ultimately bring the information from the sensors in the periphery up to the area in the brain. And since this is dealing with cutaneous sensation, it's going to ultimately go to the somatosensory cortex in the brain. Remember that post-central gyrus. And so the first order neuron, that would be the one associated with the sensory receptor. Okay. And so say there's a sensory receptor out in the skin, and that neuron would be climbing up here and bringing the information into the spinal cord. You can see in the spinal cord is where we meet up with the second order neuron. Okay. There's the second order neuron, there's the synapse, and you can see, depending on the information, whether it's crude touch sensory or whether it's, well, that would be pain. If it's crude touch, it would be in the anterior region. But you can also see what's taking place here is that second order neuron is crossing over. Okay, so information, if this was the right side of the body, that information for the right side, the sensory information for the right side, is actually going to climb up and go to the left side of the somatosensory cortex. So when that takes place, we have what's called contralateral control. Okay, contra, opposite. All right, so the right side of the body is actually controlled by the left side of the brain. So if you had damage to this part of the somatosensory cortex, you would have loss of sensation on the right side rather than the left side. Okay. So that's contralateral, and you can see that takes place because this neuron, the second order neuron, is crossing over here at the spinal cord. Right? Again, depending on the information, okay, for pain, lateral spinal thalamic tract, or for crude touch and pressure, anterior spinothalamic tract. But they would, the second order neurons would climb up spinal cord up through medulla, the midbrain, and ultimately synapsing at the thalamus. So all your sensory information, except for olfaction, smell, is going to go through the thalamus. Okay. And then that is where we're going to synapse. The second is going to synapse with the third order. And then the third order ultimately brings the sensory information up to wherever it needs to go in the somatosensory cortex. Okay, so that is your pathway there. So again, all sensory information is going to make its way through the thalamus. So it's kind of like a way station. The sensory information comes up to the thalamus, and the thalamus is going to splice the sensory information out to where it needs to go in the cerebral spinal cortex. Okay. It's not just a way station. What they're also finding out now is the thalamus has some editing information as it goes through there, but ultimately sensory information comes up to the thalamus, and then it is brought out to whatever region of the cerebral cortex that it needs to go to. Because remember, there are different regions devoted for different sensations in that cerebral cortex. In this case, with the case we were talking about, the cutaneous, we're going to go to the somatosensory cortex. So since we're dealing with cutaneous and general senses, we're coming to the somatosensory cortex. So after going to the thalamus, the information is ultimately sent to this purple region here of the cerebral cortex. Okay, and that's where we're going to get perception put on to what we're actually sensing out in the skin, in the cutaneous sensation. And what you see, the makeup of this cortex, is you have what's called somatotopy. They've mapped out this region of the cortex, and they've mapped out that different ports of this cortex are devoted to different regions of the body. So if I was to put a probe in and I was to zap you, say, right here, you would have a form of sensation out in the fingers and hand region. Okay? Whereas if I was to probe right here, you would have sensation, feel sensation out in the lips. Okay? So they've mapped this out, and that means what somatotopy is. The other bit of information you're getting from here is look at the regions, how much of the cortex is devoted to the different regions of the body. Okay, and this gives you an idea of how much reception we have at these different regions. If we have more receptors, we got to devote more regions of the somatosensory cortex to that region. Okay, because in our body, physically, our trunks is a large portion of our body physically, but you can see from a sensation standpoint, it is a very minuscule region on our somatosensory cortex. So that tells me that there is very low density of receptors, okay, neurons associated, sensory neurons for the trunk region. Whereas my hand, which is a much smaller in physical size, you can see I have a much larger area of the somatosensory cortex devoted to my hand, and that's telling me that there is much more sensory receptors and neurons devoted to my hand region, the finger, which makes sense because I have, how do I go out in the world and look for sensation? I use my hands. I use my hands, my touch on my hands, so I definitely want to have a good amount of sensation on this region. One way to determine or look at the amount or density of receptors in a specific region is we can use tactile discrimination, or what we call a 2-point threshold. And this is the ability to be able to distinguish between 2 points. Okay. In lab, if we were in lab, well, you had this demonstrated for you. If you're in lab, you would have taken some calipers, okay, and you would be pushing down with the calipers and going from small and bringing the calipers out to wider distance between the 2 points. And what you would be looking for is at what point do you feel the sensation as 2 different points. And for the brain to determine this, is it's got to get information from multiple receptors. Over here, you can see the calipers. Let's just put these calipers at the same distance between. You can see here, okay, what kind of information is the brain getting? There is this sensory receptor that is basically covering this whole area here. When we put these 2 points down, the brain is only getting sensation from activation of one sensory neuron. And so the brain can only interpret this as 1 point. Whereas here, pushing down, there is multiple receptors or sensory neurons covering this region. Okay. And now we're getting one sensory neuron firing and a second sensory neuron. So now the brain can, hey, I'm getting information from here, I can sense this as 2 points. Okay, where we have higher density of receptors, say I have this swatch of skin. All right, make this the same distance. If I have higher density of receptors, okay, the receptor field for each sensory neuron can be much smaller. Whereas if I have low density receptors, I have to have one neuron covering this whole stretch. Okay, where here I have 3 different neurons covering this whole stretch of skin. All right, so where I have higher density of receptors, you can see I have greater ability to determine 2 points in a smaller distance. Okay, where if I had caliper here, that would be 1, but with my higher density region, I can tell with this amount of distance between my calipers, I can tell this as 2 points. Okay, so it's kind of inverse relationship. Where I have a higher density of receptors, I would have a lower 2-point threshold. That means I have an ability to determine 2 points at a smaller distance apart. So the sensory nervous system, to also pinpoint where touch is occurring, it can do a process called lateral inhibition. So when we have a blunt object being pressed down on our skin, the stimulation taking place is across the whole skin, but the perception of where we're actually being touched is perceived like this in the brain. It is much more focused where the touch is taking place, and this occurs due to what we call lateral inhibition. Okay. You can see with this pin being pushed down. Okay. The sensory neuron that's being most stimulated is B here. Okay. These other neurons on the periphery also get sensation, but the amount of firing by them is much more less. You can see the neurotransmitter, the one that's right under the pinprick. Okay, that first order neuron is doing lots of neurotransmitter to the second order, where here, the outer regions, we're getting less. Even though they're stimulated less because they're not right under the center point. Okay, and so what happens is at the second order level, these are multi-neuron pathways in the sensory. The second order is actually going to inhibit those being stimulated by the other regions here. So this collateral here is basically helping turn off the signaling to the third order. So what's actually getting to the brain is we're not getting, hey, there's a little bit of touch here. We're getting the touches here. All right, because these will not be sending information to the brain because they've been inhibited here at the second order. So this is called lateral. We're going laterally and we're inhibiting this neuron. Okay, because if the information at these got information and we're firing also, we would have perceived touch would be more broad, more fuzzy. But because we do this lateral inhibition, this perception of touch is much more focused at the brain. So since these sensory pathways are multisynaptic, there are multiple neurons that make up the pathway, we can get a phenomenon called referred pain. And this refers to that certain sensory information coming from different somatic pain, it can be felt when there is a disorder in certain organs. And what is believed to be happening here is because there is a multi-neuron pathway, that pain for a certain region of skin, right? Synapses on a same neuron that also may be stimulated by pain in a certain organ in the body. So the visceral pain and the somatic pain end up synapsing on the same neuron that's going to bring information to the brain. Therefore, if I have pain stimulated by my heart, there's something wrong with my heart, it can also stimulate, or you get a referred or perception of pain in a certain region of skin because of this synapsing on the same neuron. Okay. Many times you've heard pain in the left arm. Okay, so this would be skin in the left arm, and if I had pain in the heart, I may have pain in this area of skin. And this, from a medical sense, this is important because if somebody comes in with pain, say in this region, you may want to look for problems in the liver or gallbladder, so forth. Or pain in this region, you may want to look for, hey, are the kidneys okay? Okay, and so different organs have been mapped out to basically have referred pain for different regions of the skin. So we've handled the cutaneous, which are considered general senses. Now let's look at our special senses. Okay, those are going to be the senses that are located in the head region. From our eyes, we get vision. From our mouth, we get taste or gustatory. From our nose, we get olfaction. And we'll see from our ears, we get auditory, and we get equilibrium from the ears also. Okay, but first we're going to go to taste, gustatory, and olfaction. And these sensors, the receptors for these, are chemoreceptors. They're going to respond to different either chemicals we have from our food that we're eating in our mouth, or smells, scents that we are getting in the nose come from different molecules in the air. Okay, so they are going to be chemoreceptors responding to the different chemicals either in the air or in the food that we eat in our mouth. And these, we handle these together because they are greatly tied to one another. Our olfaction greatly influences our taste. Kind of like when you go to grandma's house and she's baking those cookies. You can almost taste them. When you smell them, you can almost taste them. So that gives you the idea of how taste and olfaction are tightly wound together for that sensation. So for taste, our sensory cells, they are actually modified epithelial. Remember, we have the sensor cell is actually not the neuron, but we're going to have them tied closely to the neurons. You can see that here. You can tell which of these cells is the sensory cell, the gustatory cells, because they have the neuron associated with it. Okay, but this pink, this gustatory or taste cell, is a specialized epithelial. And what you have is you have these gustatory cells, all right, and you have supporting cells. Okay, this lighter pink, and they're arranged with about 50 to 100 of these modified epithelials arranged in this taste bud. Okay, and these taste buds are located on the tongue, and the tongue has little pores. Okay, you can see this would be another taste bud over here, and this little pores, because what do we have to do? The receptors for these are chemicals, so we're eating our food, the chemicals are washing across our tongue, those chemicals can get into the taste bud, and basically what we see is these gustatory cells have these hair-like taste hair projections, and this is where those receptors would be found for the taste. Okay, again, they are chemoreceptors because they're going to respond to chemicals that are in the food. So let's take a closer look at these taste cells of the taste bud. Okay, the taste cells or gustatory cells are those that have the sensory receptors for the different chemicals. All right. These are specialized epithelial. Remember, they are not neural tissue, but they're going to operate very similar to like a neuron in that the chemicals that stimulate them are going to cause depolarization. And once there's enough depolarization, these gustatory cells are going to release neurotransmitter to the sensory neurons. Okay, and then that will stimulate the sensory neuron. If there's enough excitatory input to the sensory neuron, that will induce an action potential and start the signaling that will go out to the brain to get perception for the taste. Okay, these taste cells, we're going to see we have 5 tastes that we taste, that we have sensors for, but each taste bud will have sensation for multiple tastes, but each of these gustatory cell, like this gustatory cell, if it responds to sweet chemicals, it will only respond to sweet chemicals. It will not respond to salty chemicals. And the same with the neurons. the neurons are not stimulated by different tastes. If a neuron's devoted to sweet, it's going to be only carrying information for sweet to the brain or in that pathway. Okay, so they're not intermixed. The tastes are not intermixed between the different taste cells, okay, and the neurons. Right? A neuron is not going to carry information for both sweet and salty. A taste cell will not sense for both sweet and salty. It will only sense for one specific taste. Okay, and what we're going to see with these tastes is they can be appetitive. Some will actually induce you to want to eat more of it, or can be aversive in that if you eat this certain chemical, you will be aversive to eating more of it. Okay, and we're going to see what tastes are considered appetitive and aversive. So we can see those taste buds, right, that have those sensory cells. They are spread out throughout the tongue region. We have the different papillae that form up the taste buds, and you can see the valate, folate here. These little speckles here are where you see the taste buds. So when you're eating food, the chemicals come in, you're chewing it up, they get in the saliva and that mixture of saliva with the chemicals in the food down the papillae and then can get into the taste pore to stimulate those taste buds. As I was insinuating, there are 5 categories of taste that we have receptors for. We have salty, sour, sweet, umami, which is a meaty type flavor, and bitter. Okay, you used to think there were certain regions that were devoted for certain tastes, but now they're discovering that all regions of the tongue have taste buds for all 5 different categories. What you will see though is taste is influenced by temperature and texture. One of those is for sweet. Cold makes the taste of sweet much less responsive. So think about those frappuccinos that you get at Starbucks, and they taste really sweet, but they are also ice cold. So you can imagine if taste sensation is decreased with cool temperatures, you can imagine how much sugar is actually put into those drinks, because you still get a nice sweet flavor from them. So again, temperature and texture are going to influence the taste sensation. So let's look at how these gustatory or taste cells are stimulated. Remember, they are modified epithelial, but they act very similar to like neurons. So they are going to get depolarized, and that depolarization is going to cause them to release neurotransmitter to the sensory neuron. Okay. For salty, what do you think of salty? Salty is sodium chloride, and so what these sensory receptors is for is sodium. In this case, the receptor that is on those little hair projections that are sticking out in that taste pore is a sodium ion channel. Okay, so when sodium is present, sodium can move in through the ion channel, through the receptor, into the cell. And what's sodium going to cause the cell to do? It's going to cause the cell to depolarize. If there is enough sodium to get enough depolarization, that will cause the opening of calcium channels, just like we saw with our neuron, right? The action potential in that case opens up calcium channels. That calcium is the cue for releasing the neurotransmitter. Okay, so calcium cues the neurotransmitter. So the sodium moving in depolarizes the cell, which causes the opening of calcium channels, and now the calcium can cue the exocytosis or release of neurotransmitter to the sensory neuron. Okay, with more saltier foods like potato chips, more sodium's coming in, more depolarization, and more neurotransmitter is going to be released. Okay, we said some of these tastes are attractive, appetitive, or aversive. Okay. In this case, salt is attractive or appetitive, meaning you want more salt at lower concentrations. At higher concentration, it starts getting aversive. Okay, too salty, it's very aversive, meaning you don't want to eat more. So sensory reception for sour taste is going to be very similar to what took place with salty. Okay, instead of the sodium ion, we have protons, because sour is going to be elicited by foods that are acidic. Okay, acids produce protons, and therefore it is the amount of protons is what's going to stimulate the gustatory cells for the sour taste. Okay, just like salty, the proton is going to move through the receptor, which is an ion channel. Protons have a positive charge, so that's going to cause depolarization of the cell, and again, the depolarization causes the opening of calcium channels in the gustatory cell, and then the calcium cues the release of neurotransmitter, and that will activate the sensory neuron. So the next three tastes, sweet, umami, and bitter, are going to be stimulated. Those gustatory cells for them are going to be stimulated a little bit different than what we saw for salty and sour. In this case, the receptors are not ion channels, they are G protein coupled receptors, and they have a specific name for those G proteins involved in taste called gustducans. Okay, and so we're going to get binding to these receptors by the different chemical molecules. For sweet, there's going to be multiple chemicals that can elicit or bind to the receptor to elicit a response. But most of those are going to be sugars. All right, those monosaccharides. And for umami, the receptors are going to respond because it is a meaty flavor, meaning proteins involved, amino acids, glutamate and L-spartate, which are present usually in most proteins, and therefore these are going to elicit the response for that meaty flavor, that umami taste. Okay, so the signaling in these are a little bit different than what we saw with the ion channels. In the ion channels, the ions moved into depolarize. In this case, binding of whatever specific molecule elicits this receptor is going to activate the G proteins. The G proteins are going to act through a second messenger system, and that second messenger system in this case is going to close potassium channels. Or like our neurons, potassium leaves and keeps our membrane potential down -70 millivolts. If potassium is not allowed to leave, that's going to cause the cell to depolarize. Again, the depolarization results in release of neurotransmitter. Okay, so that's the signaling there. So bitter as a taste is kind of unique, and it is definitely an aversive at low concentrations. So eating something bitter that elicits the bitter gustatory cell will actually prevent you from wanting to eat more of whatever food that is. Okay, many toxic compounds, spoiled food would also give you a bitter taste. So usually this is a protective response. It is aversive because it's protecting you from either toxins or spoiled food. But the bitter is also it's going to be activating a G protein coupled receptor. Okay, the signaling is a little bit different than the other two when we saw umami and sweet, in that binding of whatever bitter, there's multiple molecules, bitter molecules, but one of those, quinine, would activate the bitter receptor, and binding of the receptor will activate the G proteins. Again, will stimulate a second messenger system in the cell, and instead of closing the potassium channels, that second messenger is going to open and cause the release of calcium from inside the cell, the endoplasmic reticulum. But the result is the same in that that's going to cue the release of neurotransmitter, and that will stimulate the sensory cell. Okay, so the signaling pathway is a little bit different, but again, it is a G protein coupled receptor that is responding to the chemicals, the bitter chemicals. So now let's look at how we get the information once we stimulate those gustatory cells, how the information is relayed to the brain. Okay, because ultimately we're going to this little region of the cortex, the gustatory cortex, that's the region that is devoted to perception of taste. So we'll elicit all those sensory neurons that are being elicited by the taste buds. They are going to travel either through the glossopharyngeal or the facial nerve tract, okay, up to the medulla oblongata. Then you get your second order neuron, okay, crossing over, and you're getting brought up from the medulla up to the thalamus. Remember, all good sensory information, besides the next one we'll be talking about, olfaction, all sensory information goes to the thalamus, and then the third order neuron from the thalamus is going to take it to the gustatory cortex. Okay. You can see it crosses over, so therefore taste on the right side of the tongue are actually going to go to the left side of the brain. So it is contralateral control. Alright, so now we're going to look at olfaction. We're going to look at smell. Again, these are chemoreceptors because they're going to respond to odor molecules that are in the air that we're breathing in through our nose. Okay, so the olfaction sensory cells are going to be located in the nose or the nasal cavity. So what we're going to see with olfaction is the actual sensory cell is a neuron. So we're actually eliciting a response in a neuron in the sensory cell. Whereas taste, we saw it was modified epithelial, was a sensor cell. So olfactory is a sensory neuron. Basically, the sensory receptors that we're going to see, we're going to look at in a second, are on the olfactory neurons. What we see is there are many genes that code for different receptors. Okay, so we have sensation for many different molecules, and we have many different receptors in the case of olfaction. Okay. What we're going to see is there's one receptor cell, one type of olfactory receptor. So we're not going to be able to sense multiple different molecules on the same sensory cell. So each cell is devoted to a specific smell. Okay, and that's stated here. Signaling of cell is specific to one molecule. Okay, so say a smell that's noxious compared to a sweet smell, that sensory cell will not respond to both sensations, only to one specific molecule. So here's kind of the setup for the olfactory epithelium. This is going to be on the roof of your nasal cavity. And if you remember that cribiform plate from anatomy, remember that cribiform plate had those little olfactory foramini. So that is where these are sitting. These are those little holes in the cribriform plate when you were looking at the ethmoid bone in anatomy. Okay, so this is where the sensory neurons are going to travel through. The epithelial, the part that is going to sense, sits on the bottom of that roof. Okay, and you have projecting through there are these sensory neurons. These blue guys here almost look like a little tree with a root coming through here, but this bulb with the little hair, the little spraying roots, is where the sensory cells are. Okay, so there's going to be this mucus. Here's the epithelial cells, the layer here, and then the sensory neurons are coming through here. And they have these little hair-like projections, right? And this is where the sensory or receptors are going to be located. Okay, they're embedded in this mucus. So what happens is when you breathe in, the odor molecules get dissolved in the mucus, and then they will make their way to the receptors. Okay. Kind of unique thing here is these basal cells. These basal cells are stem cells because they are going to replace damaged sensory cells. So this is one of the regions in which we actually have stem cells for neurons, because these sensory cells are actually neurons in this case. Okay, and then we also have the supporting cells, but the actual sensory cells are these blue right here, and they run up through the cribriform plate, and then they're going to synapse with the neurons that are going to go to the brain. So that makes up that olfactory bulb you also saw in anatomy. Okay, it contains those tracts, those neural tracts that are going to make up the olfactory neuron, okay, or olfactory nerve. All right, so signaling in the olfactory cell. Right, here is the receptor. Okay, and these are G protein coupled receptors, and GPCRs. The odor molecule, right? This would be located on those little hair-like regions of that sensory cell. Those receptors, the odor molecule is dissolved in that mucus and comes and binds to the receptor. That's going to activate the G protein, and the G protein then activates adenylate cyclase. So that's going to be acting through that second messenger system of cyclic AMP. All right. And that cyclic AMP then goes and opens up ion channels for sodium and calcium. Okay. That's going to depolarize our neuron, because remember, these are neural cells, and then we can get action potential sent, and we can get release a neurotransmitter to the next neuron in the line. Okay, but odor molecule binds to the receptor, activates G protein activates adenylate cyclase, we get our cyclic AMP, and then that cyclic AMP ultimately is going to result in opening of sodium and calcium channels, and those positive ion coming in causes depolarization and activation of that neuron. Now let's look at the signaling that takes place and how the information is brought to the brain. Right, so these are the cells in which those G protein coupled receptors are located. Right, and these are the cells that are going to get activated. The sensory neurons are the ones that are going to be activated by the odor molecules. The odor molecules would be coming in here, activating. And what we see is this blue, the green, the red, they would all be neurons that are stimulated by different odor molecules. But when we come up to synapse with the second order neuron, okay, you can see that the same sense for the same molecule are going to synapse with a similar neuron, okay, in what we call the glomerus. Okay? And this is taking place, this would be the big olfactory bulb where the synapsing is taking place with the second order neuron. Okay? What's going to take place is that second order neuron is going to make up that olfactory tract, and it's going to bring the information basically back to the cerebral cortex. So remember I said all good sensory information goes to the thalamus except for olfaction. You can see the second order neuron is taking the information directly back to where it needs to go in the cerebral cortex. We're not getting the thalamus and then get splicing out. Okay. Along the lines here are also these paraglomerular cells. You can see them here going between the different glomerulus, and this forms kind of somewhat of a lateral inhibition. Remember we saw lateral inhibition allowing us to distinguish or make a better distinction between where the touch is taking place. This lateral inhibition allows us to distinguish better the smell that we are smelling. Okay. So we get signaling here. This is where we get the activation of the G protein coupled receptor. That's going to stimulate the neuron, which brings it back to the second order neuron, and then the second order neuron is going to bring the information back to the brain where we get perception of the different smells. And where that olfactory tract is taking the information, it's going to be taking it to the olfactory cortex.